Hello everyone. I'm going to talk about question 4b today. I'm going to talk about Gregor Mendel and his studies of Pisum sativum, the common garden pea, and how Mendel is given credit as being the father of genetics, and he very richly deserves that. So, question 4b is all about Mendel. So what did he study? He studied the common garden pea. This isn't some rare or exotic organism. You can actually go to Home Depot or Lowe's and buy seeds of Pisum sativum. You find this in the grocery store. We eat it by the ton. So, where was Gregor Mendel from? He was from Austria, south of Germany in Europe. He was considered a naturalist. We would now call him a biologist, but back in his day, they would call him a naturalist. And he was a man of the cloth. He was a religious man. He was a monk. He did his work in the 1860s. Ultimately, he published in 1866. Give that a little historical reference. In 1866, you think about the 1860s in this country. You had the American Civil War, 1861 to 1865, Emancipation Proclamation in 1863. So while we are over here fighting over states' rights, you have a gentleman over in Central Europe establishing the field of genetics. Except when he published, people didn't realize the implications of the guidelines and the rules that he laid out. They saw it as a study about plants. He said, oh, those are wonderful guidelines for plants. Right? They didn't understand that the fundamental principles, these rules that Mendel laid out, applied to all living things, plants, animals, protozoans, fungus. Every sexually reproducing organism follows what we call Mendelian genetics. But unfortunately, Mendel passes away and never gets the credit he so richly deserves in his lifetime. So his work sits for about 20 years after his death, until the early 1900s, and there starts to be a very serious discussion about whether the genetic material is protein or is it DNA. We discovered chromosomes, and we knew that chromosomes were actually mostly made of protein, and so biologists really weren't sure whether the genetic material was DNA or whether it was protein. So as part of this discussion, there's a British gentleman named Reginald Punnett, and he starts going to the library. This is, of course, long before the web. And in the 1900s, he starts reading the publications and the studies of the scientists that came before him. And he pulls Mendel's work off of the shelf. Now, Mendel's work was a little bit hard for people to understand because Mendel wrote it in paragraph form. And so Punnett sits down and says, all right, Mendel tells us that each parent carries these characteristics in pairs, what we now call homologous pairs, and that these are separated in a process we now know as meiosis. Mendel didn't know about meiosis in his day. Well, rather than describe this in paragraph form, what Punnett did is he drew this simple box. And he said if each parent carries these pairs, and these pairs are separated, the parent either donates the dominant A or the recessive A. And then the other parent does the same thing. You then get fertilization, the joining of these two gametes, and you get the three different combinations that can occur in the offspring. And once Punnett drew Mendel's work like this, people really started to understand it, which is why we now call this simple square a Punnett square, recognizing the contribution that Reginald Punnett made. But Punnett was never trying to take credit for the work ever. He always gave Mendel full credit. So, what did Mendel tell us? Mendel told us that there's a thing called a trait. This is a single characteristic of an organism. And commonly in high school, we present this as eye color, or we present it as hair color. And really, when it gets to the pigment of our species, the genetics isn't as simple as we present it in high school, but that's okay. Where what Mendel talked about was plant height. This was a discrete characteristic. See, in hair color, you can have dark brown or light brown, or you can have a dark red or what's a strawberry blonde. You have different shades of hair. Those are indiscrete characteristics. Whereas in pea plants, the plant height is very distinct. The plant is either short or it's tall. It's one or the other. There's also things like seed shape and seed color that are very distinct. But this is a single characteristic. It's only one, hair color or plant height. Where there's this other fun word, an allele. So what alleles are is these are the different forms of a trait. And notice here how this is plural. Okay? The alleles are always plural. There's multiple alleles for each trait. So for instance, let's make ourselves a really simple chart here. Here's a trait and an allele. So in our species, hair color, and again, the genetics on this aren't as easy as we present it. So hair color, in our species, it's brown, it's blonde, and it's red. People say, what about black hair? All right, black hair in our species really doesn't exist. If you take someone who appears like they have black hair and you take a piece of their hair and you put it under a microscope, it's actually a really dense brown pigment. Someone who has black hair is actually just a really, really, really dense brown. So this is your single characteristic, your hair color, and these are all of your different alleles, your brown, blonde, and red. But to apply this to Pisum sativum, you have flower color, and in the pea plant, the flower colors are either the dominant, purple, or the recessive, white. So this is the characteristic flower color, and these are the alleles, which are purple and white. Those are your different choices. So purple and white is where Mendel started his work. It's a classic experiment, and it's one that we get to draw. It's very cool. So let's draw ourselves a flower. So plants reproduce sexually. That's what flowers are. And what Mendel found is that the pea plants have two colored flowers. He found that some are purple. And then he found that other pea plants were white. And he inbred these plants over and over and over again. So he knew that they were what he called pure breeding. And then he crossed the purple with the white. And what he found then was that all of the offspring of these two parents, we usually don't think of plants as having parents and children, but that's how the rules work. If you cross a purple and a white pea plant, all of the offspring are purple. All of them, no exception. Mendel did this one experiment thousands of times, and we'll take a look at the data here in just 1 second. Literally thousands of times. Well, since the purple was expressed in the first generation of the offspring consistently, he then defined purple as big R. And you say, well, why not P for purple? We try not to use letters whose capital and lowercase look the same. So a capital P and a lowercase p look extremely similar. So let's use R. Well, then the recessive for white is little r. So then what happens here, and this is where Mendel explained it in a paragraph, and this is where Punnett is really useful. This parent separates these two big R's, and one gamete contains big R, and the other gamete contains another big R. The other parent goes through meiosis. One gamete contains the recessive little r, the other gamete also contains the recessive little r. These R's then come together, fertilization, and all of the offspring are big R, little r, what we call heterozygous, or a different pair, while the parents are both homozygous, or same pair. So the offspring here is big R, little r, heterozygous purple. And people had really, they had done this for years. And this really wasn't any big conclusion. But here's what Mendel did that nobody else did. He inbred these plants. He took these first generation offspring and he bred them. And what happens is he, of course, gets, well, let's make that flower prettier, shall we? Like this. He gets purple offspring, no surprise there. Aha! But the white reappear. This is where people say, oh, the white skipped a generation. The white doesn't skip. The white allele is there the whole time. Okay? But what happens is it's re-expressed in this second generation of offspring. And in fact, it's expressed in a 3-to-1 ratio. And again, we will take a look at the data in just 1 second. Well, Mendel, in paragraph form, kind of lost people here. And pundits have watched this. This parent donates either the big R or the little r. And then the other parent donates a big R or a little r. You then have fertilization, and you get a big R, homozygous dominant, purple. You then get a big R, heterozygous, purple. Down here, big R, heterozygous, purple. And then down here on the bottom right, little r, homozygous recessive, white. So when you cross these organisms in large numbers, you should see 75% purple, and you should see 25% white. And Mendel literally did these crosses thousands and thousands of times. Okay? Well then, how do you explain these purple offspring here? Well, about 25% are big R, homozygous dominant. About 50% are big R, heterozygous. And then the white are little r, homozygous recessive. So, speaking of Mendel's data, if you take a look at your image printout, and if you don't have this, then I posted it in our shared group. But in this image is what we just drew. Okay, so here's your homozygous dominant, big R. This is your purple. This is your little r, homozygous recessive white. This then leads to your all purple offspring. These are all heterozygotes, big R, little r. Mendel then crossed these offspring, coming up with your 75-25, your classic 3-to-1. These are composed of your homozygous dominant, big R, your heterozygotes, big R, little r. However, they all appear purple. They have the same phenotype. Whereas over here, you have your little r, homozygous recessive, your whites, in a 3-to-1 ratio. And then look at his raw data. He did this one cross. Get a load of this. 929 times. Just this one cross, he did 1,000 times. His data is stunning. But it's not like there was, Instagram and TikTok and Facebook and all that kind of stuff. He had a lot of time on his hands. All right, so then you go to the right. So these are the characteristics that Mendel studied. He studied 7 different characteristics in Pisum sativum. Okay? flower color, purple versus white. He did flower position on top, which is called terminal, or on the side of the stem, which is called axial. Seed color, yellow versus green. Shape, round versus wrinkled. Then pea seeds are put inside of a pod, and that pod could be inflated or constricted. These are all the dominant characteristics on the left and the recessive on the right. Then the pod colors have a different, or there's a difference in the pod color. It's either green or yellow. And then, of course, the plant itself, the stem length, the plant is either tall or it's short. But again, look at the data. Almost 1,000, 850. Get a load of this. 8,000 data points. And you go, hold it, he made that cross 8,000 times? No. He only actually did the cross, well, only, listen to me, did this cross a little under 1,000 times. But then from each plant, he would get multiple pods and thus multiple seeds. So he did the cross about 1,000 times and then collected about 8,000 seeds and counted those suckers one at a time, right? And then here's your seed shape and, of course, the data for all of the other characteristics. And then notice the ratios. This is the raw data. Is it right on 3:1? No, it's not right on 3:1. It's really close. In fact, if you look at his larger data sets, the larger the data set, the closer he gets to 3:1. So on one of his smaller data sets, smaller, it's only about 600 data points, right? 600 samples, his average is 2.8:1. But then on his large data set, his 8,000 points here, look at how much closer he gets to 3:1. You have to go to the hundredths place to get 3.01 to 1. This is a great example that the larger your data set, the closer you will get to the expected value. And so this is image 14.1. It's an excellent image out of your textbook. So all this discussion of alleles, we need to be sure that you're making the connection back to the chromosomes. Again, there's all this vocabulary that we have to be sure that you are comfortable with. So, you draw your good old-fashioned chromosome here. Boom. So, this being a chromosome, then one portion of the chromosome is a chromatid. This protein ball in the middle that holds the chromatids together is a centromere. And then when we talk about these genes sitting on the chromosome, the section of DNA is physically right there. So we were talking about the big R, fine. And the position of this gene is called the locus, or the plural is loci. So that is the locus of the gene. Let's define that, be sure that you're all comfortable. The locus is the position of an allele on a chromosome. And so this is the gene that is read by transcription, production of messenger RNA to translation. Messenger RNA is then read by the ribosome to link amino acids to make protein. So when you express this gene, you get to the physical appearance of the organism, what we will soon call the phenotype. So when we talk about the organism, the Pisum sativum being big R, we refer to this as being homo, same, zygous, pair. So this is homozygous, but it's homozygous dominant because this is the same pair and they're dominant, which means the chromosomes of this organism, of course, there's a homologous pair, and it would be the big R here, and then same locus, and happens to be the same allele. As opposed to the heterozygote, hetero meaning different or other, zygous still meaning pair. This is commonly also called a heterozygote. Still a homologous pair of chromosomes, same loci, because that's where this characteristic is carried. But instead here, this is a dominant R, but this is the recessive R. So when we write those letters inside of the Punnett square, please understand that those are actually genes that sit on the chromosome, and then we pass these on to our offspring. And you have little r, still homozygous, same pair. But it's not homozygous dominant. This would be homozygous recessive. Hey, what do you know? 2 chromosomes, same loci, except now they are little r, little r. So that's an introduction to Gregor Mendel, alleles, traits, and loci on chromosomes.